we heard a bit about DNP this morning, introduction on hyperpolarization. And yeah, today I'm going to present a bit how we use another type of hyperpolarization called photo CIDNP or photo kidnap, and how we interface it with drug discovery. Before starting, I should introduce a bit who we are, because you probably don't know us. We're a very young company. We spun off from ETH a couple of years ago. We have four founders. We have mostly scientists. We have also Anil, who is a business person. And I'm mostly now in charge of the business activities of NextMR. And most of the science is done by Matthias and Roland Rick as well, Professor at ETH. What we do, so as I told you, we do hyperpolarized NMR. We like to call it ultrafast NMR because this is our purpose, is really to accelerate, use hyperpolarization in this view. And we work mostly, but not only, with fragment-based drug design. Today, just before I start, I want to explore a bit how we think about utilizing new technologies into drug design. It's not necessarily only about the scientific and the technicality itself, but how do we approach these techniques to create some added value into these processes. That's why we have to understand a bit where is NMR sitting in most drug discovery processes. If you probably have seen this type of charts many times, right? You start from hit discovery, right, where you discover some new first molecules as scaffolds. Then you modify them in hit to lead. You then lately optimize them when they start to be very potent. And then you go into preclinical and hopefully clinical trials. NMR is very handy at this early stage when you are between the discovery of new molecules and the fast iteration loop that we call the DMTA loop for design, make, test, and analyze, where you need to identify which molecules are active, go then to the next round, select new molecules, characterize them, and try to get better molecule at each round, mostly focused on potency. One thing to understand is very often the make stage, so basically synthesis, producing material, is the limiting stage where you spend the most money, the most time. So NMR comes at the test and analyze right stage. So we need to think, how can we make sure that we have a method that is addressing these bottlenecks of a requirement of material, and then as well the speed of implementation being the feedback loop of information for design and make phase. So NMR is useful at many stages, right, of characterization, from screening, meaning identifying if a molecule is binding or not to a protein, affinity determination, and as well structure determination. So you can obtain a wealth of information. This is what has made NMR such an important method in the field and probably a gold standard. Although it's maybe not the most used structural biology technique in pharma, it still has a very high reputation in terms of the quality of its data. And of course, what you see in NMR is what happens in your sample. So this is can be a hard truth, but this is the truth. Yeah. The problem of NMR is very often linked to the poor signal to noise that we have, right, and poor sensitivity. And this has a consequence, like in any analytical method, signal to noise is not linear, so you cannot simply acquire more signal, right, and it's not linear in terms of what you gain. To obtain 10 times more signal, you need to measure 100 times longer. And this is, those things you know, right? You all spectroscopy, so you know that it's because of this difference in spin population. You can do many things, right? But reducing temperature when you do if you do study life science system, it's difficult, right? You can as well increase the concentration of your sample, but it's not infinite. You have proteins. They tend to aggregate, small molecule aggregates. You often in water or aqueous buffer. You can not do whatever you want, and increasing the magnetic field is not necessarily always possible. So this creates different types of problem. One problem is throughput, right? Especially when you want to test at hit identification stage where you have thousands of molecules to test. You can spend weeks, sometime of screening, which is not really aligned with sometimes with the chemistry timeline. Sample material. So sometimes you need to produce a lot of proteins that are difficult to express or that are not stable, and finally hardware. So you need big NMR spectrometers that takes place. Not everybody can be equipped. So you don't find NMR spectrometers in any molecular biology lab, but you find other biophysics often in that spread across different biology labs. So NMR labs have been very specialized also because of this need for specialized hardware. It's expensive and that requires some expertise to be handled. So we need to find other approaches than scaling hardware to increase the signal to noise. And for that, you have many different hyperpolarization methods. I mentioned the most known ones on para hydrogen. I put Saber and FIP together just for the sake of simplicity of this presentation. And we thought about it in a way that is not necessarily performative, right? Because if you look at the performances of Kidnap, they are relatively modest in front of DNP or para H2, right, methods. But we look at other parameters. So photokidnap has this advantage that you can perform it at room temperature, right? In solution, this is simply, I will show you, it's simply a photosensitizer and present a solution with some light. So it's really easy. While DNP and Para-H2, they have more constraints, so it's a bit more complicated. And this has created some need for higher engineering. And we've seen this morning the level of engineering that has been necessary to develop efficient DNP methods. And we're not engineers, so we needed to have something that was easy for us to implement. And as well, when we think in terms of scale, if you want to propagate this technology, having something that's easy to engineer makes it very easy for us to propagate to the users. So yeah, simply light for photokidnap, gyrotron and alike in DNP. There are also some other stuff that we've seen this morning. And for Para-H2, hydrogen polarizer are not that expensive, but this is still some implementation in your lab that you need to make. In terms of cross program, one very advantageous stuff for the kidnap is it's completely integrated in your pulse program. You can bring light at any moment. You have like literally a live pulse that creates some polarization, and you can repeat scans, and at every scan you can create some polarization very quickly. So every second or so you can generate some polarization and measure something new and have 2D NMR, 3D NMR. But there are some limitations that I will discuss a bit later. Of course, one problem of photokidnap, and this is the first thing that we focused on, is the chemical space, right? So DNP is, in principle, not limited to any molecules. Although we saw that there are some considerations with radicals that have been used, but you could apply it to many molecules without limitation. Para-hydrogen is not really discussed in the field, but there are some limitations as well due to the catalyst and the steric hindrance. But for photokidnap, it's really limited. We did not know much of what we could do. So what's photokidnap? Really quickly, I don't want to dive too much into that. As I told you, you have a molecule that is in solution, your molecule of interest, let's say tryptophan. You add the photosensitizers. The most known is probably flavin, but recently there's been fluorescein, ethyl 12, a bunch of other photosensitizers. You shine light at your sample, and you have hyperpolarization. Within a second or a couple of seconds, you can get signal enhancements in the range of 10 to 50 signal to noise enhancement. So yeah, huge gains already in terms of time, that you say, because as I said, 10 times signal enhancement is 100 times faster measurement, which for us was enough. And I don't dive into the photokidnap mechanism here because we don't have time for that, but I want to leave you with maybe a couple of take-home message. You have mainly two things that are important in photokidnap to get some hyperpolarization. The first one is you need to create some radicals. Okay, so you need to generate that your photosensitizer will be excited by the light and will generate a pair of radical with the molecule of interest. And this is basically driven by Marcus theory. Which is the equation that you have here, and we don't need to dive into that, but what's important that you then need to understand, to know the orbital overlap between the two molecules, the reorganization energy, this is energy between the conformation of the ground state molecule and the radical, and some ionization potential and electron affinity, right, for the electron transfer. The other thing is the hyperpolarization is driven by the hyperfine couplings, which drive the spin sorting, right, between the alpha and the spin state. And these hyperfine couplings, so you need to have, this is the hyperfine couplings, they are creating a delta in the processing frequency. And this depends on the nuclear spin state. So if you have some alpha spin states, right, it will spin, the frequency will be less than if you have beta spin state. And this depends on the sign of the hyperfine coupling. So higher hyperfine couplings, more spin sorting, more polarization. And you see there's as well a dependency between the G factors of the electrons and the magnetic field that we're going to discuss a bit later. My point here is not those equations, is how do you obtain these parameters, okay? Well, to obtain these parameters, you need to run DFT calculations, okay? You need to run many different DFT calculations, and they contain error, they are computational heavy, and now we want to make libraries of thousands of molecules, because before that we had only a couple of molecules that we knew about. So started to be a bit heavy, and the price of simulation compared to the accuracy that we were expecting to obtain was very high. So we approached it in a bit different manner. And why we approach it in a bit different manner is because we were starting to do that with tyrosine tryptophan, like everybody else, get our signal to noise enhancement, and we started to have some hyperpolarization for some impurities, and this hyperpolarization was much better than what we had before. So we started to think, okay, this is possible to get photohyperpolarization or photokidnap for molecules beyond this chemical space. So what we did, we started with tryptamines because they are close to tryptophan. So we took LSD, amylin. This is present in ayahuasca, which is another psychedelic drug. Melatonin, serotonin, and we started to do photokidnap. So the funny thing was we took probably 6 months to just order these molecules because we had to file a lot of forms and get a lot of custom issues, and this measurement took literally 2 seconds. So yeah, not very smart of us actually to gain in terms of throughput. But what we learned was different scaffolds. Okay, we can propagate that, but it's not enough, right? If you look at fragments that contain this type of features, right? If you take phenol and indoles and imidazoles from histidine, it's only 5% of the fragments that you may find in KMBL database. So we started to look where can we find new aromatic scaffolds that we can decorate to make fragment libraries. So we took it in a very brutal manner. We find people who had libraries that were ready to open them for us, and we went and we screened compounds by thousands and started to identify aromatic scaffolds. And now we have about more than 100 aromatic scaffolds, and not to date, I think 120 something that we can decorate. And actually in any fragment library, in average, you have 30 to 35 aromatic scaffolds that are decorated in very different manners. So we have more than enough to make chemical libraries now that with sufficient chemical diversity. And this was at the cost of testing about 10,000 molecules since, not 2025, sorry, since 2022. Yeah. Now we have a library of about 500 fragments that we maintain physically. We have up to 5,000 that we have in our database that we don't order because we're a small startup. We don't have the money for that. It's quite expensive. But we also have a very good collaboration with ChemSpace, which is the biggest catalog of molecules, and you can generate molecules through Synton technology. So you can enumerate molecules, billions of molecules, without a lot of computational power. And now we are developing a lot of surrogate parameters, meaning that they don't require DFT calculation, that help us to identify photokidnap molecules very quickly. And we expect to have about 30%, 25 to 30% of the chemical space that will be photohyperpolarizable with photokidnap. So yeah. The other thing is now we measure compounds within a couple of seconds. We have thousands of compounds and we need to measure them. And the thing is, before that, it was done with bringing an optic fiber into an NMR tube. And we created a new problem that was good for me at the time. I had a few more kilos, and it helped me to lose those, because you have to literally do that hundreds of times. And I think this is a video when I was in Latvia screening compounds, where I think we screened up to 300 something samples in one day. So it was just literally inhuman. There was COVID lockdown. I mean, we had nothing else to do. But the thing is, we needed to get out of optic fibers. Some people have done some effort. Actually, in Manchester, Sasha Gorvanov has developed this NMR touch technique that is quite nice because you can have a better illumination. You don't need to have an optic fiber. But the problem is still throughput is not easy to scale in terms of throughput. Until we realized that an NMR tube is actually an optic fiber. If you bring light here at the bottom of your NMR tube, you have some lens, right? It refocuses the light, and then the reflective indexes of liquid and of the glass around the NMR tube contains the light into your sample. So Of course, illumination is a bit less homogeneous, but for us, it was not, we found that it was not an issue. So we could now bring the optic fibers from the bottom of the cryoprops because you have this air conduct, and now you are able to sample, to bring your sample with a robot. So it works with any robot here. It's a sample mail, I think, and then we have this as well with another sample jet, and it's very convenient. So we have on the sample jet now about a throughput of about 500 samples per day. We can go up to 700 samples per day when we remove all the safeties of the sample jet. And we do it sometimes, but yeah, it's not ours, so we have to comply. But we are very low error because of course it's all fixed, so we don't need to have we don't have position of the optic fiber in the tube that changes. The point is, I think we will reach probably in the next 2 to 3 years about maybe 1,500, 2,000, maybe a bit more than 2,000, mostly by optimizing how robots insert samples and shimming protocols, because we have always the same sample on the screen. So we can probably optimize a bit to things and gain a few precious seconds that will help us to multiply a few times this throughput. Yeah. Finally, I told you there's something quite important, which is we have big NMR spectrometers, but there are also some small benchtop spectrometers that are actually quite cool. And this advantage, we thought about it because when you look at this equation, you see that when you reduce the magnetic field, you are supposed to increase the polarization and the efficiency of photokidnap. And this is, when you look at this relationship, this polarization now is, you have to look down. I don't know why the scale is disappeared. Anyway, so it's 0 to 1 in this direction. And you see that when you reduce the magnetic field, it's decreasing unless you have a very tight delta G. So you see, if you have a tight delta G, then you will have, at some point, it will bounce back because this equation is actually a very simplified version only for this part. But most of the time you have a relatively big delta G, so reducing the magnetic field is quite advantageous. We verified that using this is data on the Fourier 80, where we could see that, you see, the signal to noise enhancement was improving. And why I mentioned the delta G is because it improves depending on if we had a small or bigger delta G between the molecules that we tested. So here for tryptophan and fluorescein, we had a small delta G, so we got a modest improvement by going to 80 megahertz. But for this one, we get a bit better, like a tenfold improvement in the signal to noise enhancement. And for this one, we got something quite crazy for kidnap, is that we got about 1,000-fold signal to noise enhancement for this type of molecules. So it was quite impressive to be able to do that. So now, measuring a lot of molecules very fast, how do we do drug discovery, right? How do we use that to make sure that we can identify molecules? And this is all luck. I mean, we had some luck with the magnetic field, but another luck is that when you hyperpolarize molecules, okay, this is a very bad analogy for NMR spectroscopies, but I mostly show that to chemists. So you are hitting the molecule, which is thermodynamically false, right? But anyway, just accept my very analogical approach. You heat up your molecule, and it's like a hot glass of tea, right? This is out of equilibrium. You don't hyperpolarize the protein with photokidnap in the regime that we use. I can go into that a bit later, or if you have questions about that, we can discuss, but just accept this fact. We don't hyperpolarize the protein here. And now the protein is like a bucket of ice, right? So when your small molecule interacts with the protein, it just cools down, right? It's like dipping literally your cup of tea into a bucket of ice, and it cools down. And this is basically cross polarization, what is happening here. So just the fact of measuring a simple 1D, hyperpolarized 1D with and without protein is enough to see if there's an interaction between the molecule and the protein. And this is very fast. We can do a simple 1D. The other thing that we can do is that we can do other normal experiments, right? Other more classical experiments like a CPMG to detect a T2. This was scaled here, so you can obtain here the T2 relaxation. And it works as well for 19F. So for 19F, we got cross-polarization effect and the T2 classic effect from the 19F, or even sometimes some chemical shift that you can already observe, where we were able to observe that. So this is very handy, and a CPMG takes us about 10 seconds to record the decay for the CPMG, and we have as well the NOE for the first point without delay. So very, very easy. So yeah, about a few seconds to measure the experiment. We can integrate the 1D quenching and the CPMG or the echo within this. It's quite interesting for us because in drug discovery, we like to de-risk a bit the molecules that we measure. So here we have two different processes. We have spin density, right, because of the NOE effect. And here in the CPMG, we also have the we measure with the low frequency, so we also have the chemical exchange. So we can validate a bit the molecules already and before going to orthogonal biophysics like SPR or MST. Another thing, it's quantitative. It's NOE-based effect. So in T2, you always have this exchange, which is a bit annoying to deal with. And it's very analogous to STD-NMR, if you know it. So you can do simply a ligand titration where you increase the ligand concentration, you maintain your protein, so with and without protein again. And within very quickly, you can measure your buildup. And similarly to STD-NMR, you create your amplification factor that is proportional to the KD, right? That you can then fit within the KD equation. And what takes usually a couple of hours or even a bit more sometimes now takes us about 15 minutes with a few micromolar of protein, let's say 2 to 5 micromolar of protein and a bit more of the ligand. I mean, as much as you need for the titration. Finally, 2D NMR, right? I mentioned like, okay, we need we can do a bit more complex experiments very easily, and we could do eventually as well 2D NMR very simply and many different experiments. Kidnap has one issue. You are shining light at the photosensitizer. What happens is what you can expect. It bleaches. Okay. So this has been a problem for us, a limitation in the complexity of the experiment that we can measure. But recently, we figured out that we could add some adjuvants that reduce considerably the photo bleaching. So we could measure, I think we expanded that for about up to 200 seconds. This is at the price of a little bit less signal to noise enhancement, but we are now able to measure 2D NMR without having this bleaching problem. What we do is we do mostly noses with that. It helps us to figure out, for instance, for free and for bound ligand. So this is, you can track the sign change in the presence of protein, which is a sign of binding of the small molecule. So it's a validation point without having to use an HSQC, for instance. If you don't have a lot of protein, that's quite useful. And also you can get the structure of a free and bound ligand. Likewise, you can do in-pharma experiments. So when you have a lot of hits, you may not be able to obtain the full nosy structure. And in NMR, often, even with techniques like NMR2, sometimes you don't get the protein ligand structure. But with in-pharma, you can really, for instance, validate docking results as well with X-ray crystallography. We work with X-ray crystallography companies where sometimes you don't have the molecule does not fit into the crystals because of crystal packing effects. And then you're able to place other hits because now you have these restraints, this network of restraints between the different small molecules. So ENOEs are very useful, but you could as well get ligand protein NOEs to do some protein ligand structure calculation. Again, what used to take us about a couple of days is now taking us 30 to 60 minutes to measure. So quite some acceleration. So again, this is why we call it ultra-fast NMR. We're quite conservative, so we don't call it high throughput because high throughput would be 100,000 samples per day, the real definition of high throughput. But I know we tend to be a bit loosey with that, but we like to call it ultra fast for that reason. And it's about all this suit that we accelerate. To show you a couple of examples how we implement that. We started with pin1 protein because we studied it a lot. It was the first library that was about 200 compounds. And we screened it. We found 20 hits. And what was quite interesting is that we got 20 validated hits. So no false positives. The reason for that is not that it's much better than the rest. It's just that we're screening at a low concentration. So we had about, we did the first screen at 50 micromolar, but then the second screen was at 10 micromolar of ligand and 2 micromolar of protein. So this enabled us to remove some artifacts from STD-NMR, for instance, from CPMG, is when you have the small molecule that starts to form some small aggregates or some small micelles that are often causing some of these of artifacts, but they are linked. This is linked to the concentration of the small molecule. So if you reduce the concentration, you inevitably reduce the amount of false positives. Of course, I'm not saying that this never happens. It was a quite small data set here, 200 fragments on a lot, but it's still already very encouraging. We did some affinities that we validated against HSQC titration to make sure that we had the right one. Another interesting thing is, you have this signal enhancement and the atom assignment, right? So the cool thing is that the quenching, because it's NOE based, is proportional to the spin density in the binding pocket. And Isabel Krim, for instance, from has been doing a lot of stuff like this about epitope mapping, so how the compound is positioned. The thing is that this is for free. You measure your 1D and you already have these parameters. You have already for each atom what is the quenching associated to each proton. And then it gives you some indication. You can use that, for instance, to validate some docking results or to check with your X-ray structures. Is it matching your data? For instance, if you have a doubt about some positioning, sometimes in X-ray the spin density, for instance, is not enough. So the electron density is not enough. So you don't know, especially for fragment orientation. It could be both orientation. So this can be a way for them to improve the data sets. So you see a lot of data augmentation that you can do at very high throughput and very low cost. Right, of data analysis. We repeated this study on the benchtop NMR spectrometer. Here, the cool thing is, benchtop are better than high field magnets when it comes to screening with photokidnap. And I don't hold my statement because I'm absolutely convinced of that. We could identify some new hits that we could not see by measuring exactly the same duplicate sample on a 600 megahertz. The reason for that is because you improve the hyperpolarization, okay, and you reduce the base polarization of your protein. So you have then a higher delta, and it seems to be working very, very efficiently. So we could validate the new hit that we found with HSQC. We always validated at this stage with HSQC, and I think we could go down as well to really low compound concentration. So I think we kind of claim that this 100 nanomolar concentration was detected, but the reviewer asked us to to calm down a bit. So we had 250 nanomolar. They accepted that this was actually detected. But to give you an idea, to measure this concentration on the benchtop without photokidnap, it took us 3 minutes for the 250 nanomolar. Without photokidnap, it would have taken 90 years if you have to respect all the D1 and everything. So you see, acceleration. Not really useful for drug discovery at this stage. It's very, anecdotic, but it shows well that you don't necessarily need huge signal-to-noise enhancement to already have huge gains in terms of time. I saw there was a lot of protein discussions, peptide, macrocycles, so I decided to just show you a bit that we don't do only fragments. You can as well do that with peptides. You have aromatics, amino acids, or non-natural aromatic amino acids, and we can do a lot of stuff. So we've been doing, for instance, KD titrations. We've been using them as well as probes for competition. And I don't put the equation to show off, but it's just because you see it's quite complicated. And when you do competition, you have error dispersion that propagates. So you have to be a bit careful. But this is very useful for ranking. For instance, if you have your probe and you want to rank different type of competitors, using that is very efficient. You have a very fast readout within, again, a few seconds. Lastly, I want to talk about a couple of targets and how we try to integrate our platform into normal classic drug discovery workflow. We work with PGK1. At the beginning, I was very skeptical because PGK1 is part of glycolytic cycle. So I was like, okay, it sounds like a biochemical weapon more than a drug, but they convinced me I was wrong. PGK1 could be very useful in cancer because you have Warburg effect where you increase the energy production of the cancer cells. So we targeted therapies like Protax or Riptax. If you're familiar with that, you could be a bit more selective of the cancer cells. As well, PGK1 is very interesting because if you increase the activity of PGK1, now not inhibiting it, but increasing it, it has been proven to be very important protecting factor in Parkinson disease models in mice. So we started to decide, okay, let's see. And NMR will be very useful because if you want to look for activators, inhibitors for different states, it's very useful to have something in solution that you can easily screen with at different states that's not X-ray. So we started screening, and what we did to accelerate that a bit is that we started to do docking. And we then started to focus our screening. We identified a quite good hit at low PGK1 concentrations. This is 10 micromolar of small molecule, 1 micromolar of the protein. This is pretty much our standard, 1 to 2 micromolar of the protein. And then we could get the KD for it. So yeah, quite good KD. IC50 as well was quite good. And then we compared the binding poles in the docking with ADP binding. It was overlapping, but also with terazocine, which is an activator of PGK1. So we see that it was overlapping, and we think that why the mechanism of action, maybe the terazocine opens the binding pocket of the PGK1. And it's very not understood mechanism, but we're looking for this type of molecule that will increase activity. Unfortunately, in our case, it decreases activity, but it helps us to understand a bit more which part may be important. So maybe the part that is important in terazocine is probably the part a bit outside of the binding pocket that may anchor a bit the dynamics of this binding pocket. All right. Quick point about integration with other methods. What we realized is that when we screen everything, you see that we have pretty much in green, these are the hits that we found in photokidnap. We had absolutely no correlation between the docking scores and the and the photokidnap. It's not to bash on docking. It's just to say like, okay, probably the way to go is that you need to have already some experimental basis, then dock on more advanced molecules that have a bit more that are a bit bigger to have better scoring functions. And also one thing is that it's not really advantageous to take a library of molecules, screen it in silico, and then order those molecules and buy. This is very expensive for us. So maybe for some bigger corporate companies, this is approachable. This is an approach that can work. But for small companies or maybe for small academic labs, you don't really necessarily gain a lot of money. It's better to have a basis and then go a bit more at the lead stage. So now we use docking again, but at the lead stage to identify more molecules. My final example will be about the androgen receptor. It's a collaboration we have with company AI Affinity, where we are trying to find molecules for this androgen receptor that has a big disadvantage. It does not express. Okay, so this is HSQCs at 30 micromolar. I think it took them like 6 hours or 8 hours per HSQC. And it degrades pretty fast. So actually, maybe it even degrades within the time frame of your HSQC, because from day 1 to day 7, you have almost nothing. And this is with like 5% glycerol. This is very heavy. It has a big IDP domain from the gray part here and the blue part here, DNA binding domain. And there are some molecules targeting the DNA binding domain, but it's not so specific, right? So we need to find a way maybe to get some molecules binding to the IDR, to the inducively disordered region. The blue one is the one that you express. We don't express the entity. I mean, just a small part of the entity. All right, so what we did is that we have now a bigger library. We started to screen about 500 fragments. We found 52 hits. We got epitopes. So C-bind is the 1D kidnap binding, and Q-bind, if maybe you heard of it, is when you do T2, is a parameter that you calculate from T2, where you do the ratio of the ratio. So we have the two parameters, and we map it onto the molecular structure to get some epitopes. And they have a machine learning algorithm that takes epitopes to infer new advanced lead molecules. Why is that interesting for IDR? Is that you don't have a structure of the protein, right? You cannot do docking on the IDR. So even ensemble predictions is really challenging. So we use that, and using the epitopes and the machine learning capabilities, we've been designing new compounds that I cannot show you unfortunately, but we got a gain in affinity of about 1 log to 2 logs. So we have now hundreds of micromolar binders that we validated in MST, in HSQC. We could see that this is binding to some of the IDR residues, that this is inducing some shifts there. And now we have been recording the epitope, the Q-bind, the C-bind. So Q-bind is in red and C-bind is in blue. And we are now mapping this again to the lead molecules, and we are going for second rounds that is ongoing right now. So to again get some affinity. So this is a nice approach that we are validating for IDRs and difficultly targetable proteins, where you don't necessarily have information on the protein side, but you can get some information on the ligand side. Yeah, to finish, I will finish maybe with a summary and outlook, and to see a bit at the future. I ask to some young people, some that Vladimir who is here today, and how can we envision a bit this platform to integrate further in pharma workflows and in drug discovery workflows. And we've been cracking a few problems, right? We've been cracking the problem of measuring faster. Now we measure faster. We have an ongoing program, a grant with the School of Engineering of ZAV, where we develop machine learning algorithms to analyze the data a bit faster, because data analysis is a bit, now it's taking us a lot of time. We generate hundreds of data points per day. You need to have some good data workflow over there. Then we generate parameters such as the C-bind, the Q-bind, and many different other parameters that I not described today. We can get some structural characterization data. And then classically, we would integrate that into QSAR models to do some heat to lead processes. The thing is, we need to have a very, very lean workflow, because you need to be perfectly integrated in pharma. And now we're talking about using benchtops. Okay, so we see really that in the future, I'm quite positive that maybe in 1 year to 2 years, you will see that you will have benchtop commercially available, integrated with light and with this type of data workflows. And that will be not only usable for NMR spectroscopists to free a bit the higher field instruments, but as well by molecular biologists who don't have this expertise, right? Because benchtop are very easy to use and to implement. So you need to have something that is very lean, and then that to have data format that integrates into these QSARS models, and maybe even in the future to address one problem, which is when you go from these NMR parameters to these QSAR parameters, right? Those endpoints, you have some interpretation levels, right? And machine learning could be pretty good at that, of interpreting directly these NMR models and these NMR data into some QSAR workflow to avoid this loss of information that we have when, for instance, we go from its binding, is it a hit or is it not a hit? The affinity has some error, and each affinity on each atom is sometimes different. We don't really know why, but this is still some information that we can use, and that, for instance, we use actually with our collaborators at Affinity. So this is where we're going, more integration. To develop this type of technologies, we have an approach that is very lean. We try to avoid whatever is very heavy in terms of development and thinking, because we try to have very, very fast iteration loop. And my final message is like that for the kidnappers, this amazing thing that, we go in the morning, we test 20 compounds. 1 hour later, we have already our hypothesis was false, but we test it 5 times in the day. And this very fast iteration has been a very strong driver of building this platform. I have to thank a lot of people, but I guess you want some coffee and maybe some cake. So yeah, I will just thank you all for your attention.